And so I'm delighted to invite Dr. Mark LEVINE. Mark is a Professor Emeritus in the Faculty of Pharmaceutical Sciences and Chair at UBC Children's and Women's Research Ethics Board at UBC. Today, Mark will discuss why it takes so long to develop treatments and vaccines. And Mark, I invite you to take over the screen.
Thank you. Thank you, Aideen. Let's see. Sorry. There we go. So, hello everyone, and thank you very much for joining us today, and thank you, Aideen, for that introduction. This is the first lecture I've given on this platform and first session that I've had. I'm sorry that I can't interact with you during the presentation and not in direct contact during the question period, but please, I hope you enjoy it and I hope you get some useful information from what we're about to talk about. And we have a chance to answer some of the questions that you may have on your mind. So the title, Why It Takes So Long to Find Treatments and Develop Vaccines, for, in this case, for COVID-19, is kind of an ironic title because in fact, things are moving incredibly quickly, and I'll hope to try to convince you of that during the presentation today. Perhaps things are not happening as quickly as people would really like them to be, but in the scale of things as they usually occur, it's happening incredibly fast. So the outline is as follows. We're going to talk a little bit about the SARS-CoV-2 virus and the COVID-19 disease that it causes. I'm going to ask the question and try to answer why viruses are difficult to treat. We're going to talk a little bit about repurposing drugs for COVID-19 and what that actually means. We're going to discuss whether or not it might be possible that we will have new drugs for COVID-19. Not likely, and I'll hope to explain why. Then what will it take to have a new vaccine or drug? What are the requirements it takes to bring a new vaccine or a new medication to market? And what are the issues that we face with regard to a vaccine for this virus? We then will discuss some of the questions we're going to have, even when we do have a vaccine, if we do have a vaccine. What are the questions that will remain at that time regarding vaccination for COVID-19? These are going to be important to consider when we get to the stage that we can actually use a vaccine. And if we have time during this part of the session, we're now in the process of beginning to open up the economy in the absence of a vaccine. What are the implications of that, and what are the questions we need to think about, all of us, in the process of opening up in the absence of a protective vaccine? And then hopefully we'll have time for your questions and get into some discussion. So that's what we're going to try to do in this session today. So SARS-CoV-2 is the virus, the new coronavirus, the novel coronavirus that everybody is trying to protect themselves and their loved ones from. This is what the virus looks like, and you probably have seen pictures, schematic pictures like this before. On the left-hand side is the outer coat of the virus and the spike proteins that protrude and make it look like a corona. And it's the spike proteins that you see sticking out from the envelope of the virus that is currently the target in most cases of the vaccines that are being developed as candidates. If you cut into that virus by cross-section, you can see that inside the virus is the coiled RNA. The RNA contains the genes for this virus that determine what the virus consists of and can make copies of itself. So this is an RNA virus, not a DNA virus. Some efforts are being made now, and I'll try to explain why, to develop vaccines that will use RNA rather than DNA or DNA as a means of vaccinating the population. So virus genes are retained there in the RNA. Just remember that for later discussion. COVID-19 is actually the disease caused by this SARS-CoV-2 virus. And you've seen pictures of people being screened at airports and elsewhere. This came about in March. And so the disease we're trying to prevent is COVID-19. And in this talk, when I talk about the virus, I'm referring to the SARS-CoV-2 virus. When I talk about the disease, or when I mention COVID-19, I'm talking about the disease that this virus causes. So the next step is to talk about how viruses actually work and why they are so difficult to treat. So what you see here is a schematic diagram of a host cell. This could be a cell lining your respiratory tract. It has an envelope or a membrane around it. And in this case, there's a coronavirus particle adhering to the surface of that cell. And the spike protein that you saw in the picture a moment ago it enables the virus to attach quite strongly to receptors or mechanisms on the surface of cells that allow them to gain entry. And so when the virus is able to get inside the cell, then it can begin to do its work. And viruses are on the, we should say, on the borderline of living things. They cannot live external to host cells. They require a host cell to make copies of themselves. So once they're inside, the virus will open itself up, release its genetic material, which is RNA, and some of the other proteins that accompany it to help it get started. The virus will then really hijack the machinery of the cell itself. And by hijacking, I mean make use of the machinery inside the cell in order to make many copies of itself. It'll make copies of the RNA and make copies of the proteins necessary to form new virus particles. The new virus particles will develop inside the cell, and then they will begin to leave the cell. Sometimes the cells die for some viruses, sometimes they don't, and the viruses bud off from the cells. And as these virus particles leave the cells, they're free to infect other cells to which they can adhere in the respiratory tract primarily. And so that's how viruses work to infect cells, make copies of themselves, and then infect other cells in a growing sort of chain reaction. And why they're so hard to treat is in part because the viruses use the machinery of the cell itself to make copies of themselves. So were we to make drugs that are very harmful to the virus, they might also be very harmful to our own cells by blocking the very machinery that the cells need to survive and thrive on their own. So what we need to do is develop drug strategies that can target those very specific enzymes and other proteins involved in the replication of the virus itself or in the binding of the virus to the cell surface. And that limits the possibilities. So that's one of the reasons why it is difficult to treat virus infections once they begin. So in terms of treatment, what does it mean to repurpose older drugs for use in treating a particular virus like the SARS-CoV-2 virus? So repurposing of older drugs means that we look at drugs that are already on the market or have come close to being put on the market, and a lot is already known about them. A lot of the preclinical and clinical research has already been done, so we understand a lot about these drugs. And some of them, if they're already on the market and have been for some time, we know a lot about them, and so we can avoid all the time-dependent processes to develop a new drug and just go into fairly large trials of these well-known drugs to see whether or not they can treat a particular illness, in this case, CoV-2. So one of the first drugs that was thought of as a possible repurposing drug was hydroxychloroquine. It's been in the news a lot until it turned out that it was not likely to be very helpful, and then suddenly we didn't hear very much about it anymore. Hydroxychloroquine is actually still being studied. It's an anti-malarial drug, and it also is used to treat a number of immune-related diseases and has been on the market for quite a long time and is effective for those treatments, but there was really no good evidence whatsoever that it was effective in treating COVID-19. There were sporadic cases discussed and interest mentioned by clinicians and others in the field that led people to believe that it might be effective. And that's a good reason to start a clinical trial. But the clinical trials that have been done were not very promising. And in fact, in one case, in a trial in France, it turns out that this drug is quite toxic. And this toxicity was already well known. It causes an abnormal rhythm of the heart, which can be dangerous. So it looks like hydroxychloroquine, although it's still being looked at, is probably not going to be a useful drug for treating COVID-19, although that still remains to be seen. The one we've all heard a lot about in the last week or 10 days is remdesivir. And it is an older drug in that it's been around for over a decade, but it has never, to my knowledge, it's never been approved for use in treating any particular virus. It had been tried in Ebola virus treatment, and it had been tried in the earlier coronavirus pandemics with SARS and MERS viruses. And it does have fairly broad antiviral activity against RNA viruses, which is why it was being thought of as a possible treatment for SARS-CoV-2 that causes COVID. And you probably heard last week that that they stopped the trial on remdesivir because it looks as though it does have an effect on COVID-19. This has not yet been published in the literature, although there was a previous study done in China published in the Lancet, which is a major medical journal, that in that study, it looked as though remdesivir doesn't do very much at all. In the current study in the United States, run by the National Institutes of Health and by the company Gilead that manufactures remdesivir, it looks as though remdesivir may shorten the course of the infection in people who are hospitalized by several days. And that was a significant finding in that study. And while the mortality rate in the people treated with remdesivir was lower than that in the people who were controls, who presumably received placebo, there were insufficient numbers of those cases to determine if that was a chance effect or if it really was caused by the remdesivir. And in fact, the people who made the final decisions on that trial, they stopped it short because it looked to be effective for shortening the course of the infection before they had enough people to see if it would reduce fatalities. So it's unfortunate we don't have that information, but we now have one very positive study and we have one negative study. So does remdesivir actually work for COVID-19? Maybe, we don't really know, but it has been given emergency approval now by the FDA in the United States, and it is going to begin to be used. One of the challenges is going to be that that we don't know if there are going to be enough doses to treat everybody who would be a candidate for this drug. And so physicians are going to be in a position, I think, where they have to make some difficult decisions as to who to give this drug to before we have enough information to know. So what about new drugs? Why are new drugs not likely to be developed? So what I'm saying is they are not likely to be developed in the near future for COVID-19. And why is that the case? So to understand that, we have to look very briefly at the drug development process. For a new drug for a disease like COVID-19, you have to start in the preclinical stage to say, well, what is the target? What part of the disease are we actually trying to treat with this drug in a way that we think will either prevent the disease, modify the symptoms, or perhaps even eradicate the disease? And that takes a lot of preclinical work, laboratory-based work, and eventually animal-based work. Once you think you have a compound or a number of compounds that could affect that target, what happens is the pharmaceutical companies will then manufacture thousands of these compounds and begin to look at their chemistry to see which of those might be good lead compounds. So they will study the chemistry and the activity against the target using techniques in laboratory, in tissue culture and in cells. And they will, from those studies, identify some potential targets, some potential medications, that they can then try out in animals. And they will have to do a number of studies in animal models to see if the drug, if the chemical is actually druggable. That means can we get it into the body? And does it seem to be safe in these animal models? And how is it removed from the body? Does it cause any toxicities? That all takes time. And once we know that, we may have a couple of compounds that could actually proceed to clinical trials. And we need at least 3 clinical trials, types of clinical trials, before a drug can be approved: phase 1, 2, and phase 3. The phase 3 trials are the final, large, usually multi-center clinical trials that are considered the pivotal trials to decide whether or not a drug can be approved to come to market. And then filing can be made and the regulatory authorities can agree, and then they can give market approval. And with any luck, after 15 years or so, one compound is approved and released to the market. So while some of the early work has already been done for this virus and the COVID-19 disease, we're not likely to see a new drug in the foreseeable future because of the time scales involved. So that leaves us with the question of vaccines. So what is the purpose of giving a vaccine? The goal, of course, is to vaccinate us with some component of the virus or something in the virus or something that relates to the virus that will cause us to activate our adaptive immune cells. And we have 2 major components of our adaptive immunity. We have B cells that produce the antibodies that you've heard about. And those antibodies can hopefully block an infection when a virus gains entry into our body. If the antibodies are circulating and present, they can bind on to the virus and prevent it from gaining access to the cell that it needs to get into in order to begin an infection. And if some of the virus particles do get inside the cell, we also have T cells. And these T cells are part of the immune system, and they recognize cells that have been infected by the virus, and they can attack and kill those cells before the virus has a chance to replicate itself and go on to infect other cells. So the goal of vaccination is to try to prevent infection or prevent disease when infection has occurred by mounting an immune response against the virus. So what does it take to bring a new virus vaccine to the clinic? It's a very complicated process, and this one is laid out by the GlaxoSmithKline company, and they're the ones that have developed the new HPV vaccine. Well, it's been on the market now for several years as a means of trying to prevent cervical cancer in women. And they show their process, and I just draw your attention to the bottom of the slide first, and you can see the timeline of developing a brand new vaccine for a disease that does not currently have a vaccine, and the whole process takes 10 to 15 years. And that's because we have to start with some of the preclinical work, some of the early work to identify what part of the virus do we want to target that might actually help neutralize the virus if the immune system can attack it. That takes some time, and the good news is that technology has improved so rapidly that within weeks of the outbreak in China, when it became publicly known, the Chinese had already sequenced the the RNA genome of this virus, and a new science of bioinformatics enabled those scientists to determine what proteins are produced by this genome of the virus and what do they do. And that has led to a huge amount of knowledge in a very short period of time that would normally take years in the past to identify possible antigens that could be used to develop vaccines. So that's already been done. And before a company or a research enterprise can actually decide to develop a vaccine against those targets, it needs to decide what kind of vaccine it wants to develop. So we need to know a little bit about the major types of vaccines that can be tried. The common ones are these: live attenuated vaccines. These are vaccines that have live virus, but the virus has been attenuated, and that means that its virulence has been greatly diminished, so that it can cause an infection or get into cells and replicate themselves, but it no longer can cause disease. And those of you who've taken the new shingles vaccine have been exposed to a live attenuated vaccine, and these are quite good vaccines that develop a fairly robust immune response. We also have an inactivated or killed vaccine. So these are whole virus particles that have been treated so that they cannot cause an infection at all, but they're still mainly intact so that they can stimulate the immune system. Those of you who get annual influenza vaccines are receiving, in most cases, an inactivated or killed virus vaccine every year. And there are a number of reasons why we have to do this every year with influenza, which may come out later. Then there are vaccines that are made out of just subunits of the virus, proteins from the virus or components of the virus that are immunogenic. That is, when we give them to animals and to people, that can mount an immune response. And those of you who travel or have traveled before the pandemic probably have taken the hepatitis B vaccine. And that is a protein subunit type vaccine that enables us to gain some immunity against hepatitis B. The newer types of vaccines are nucleic acid, which are RNA and DNA vaccines. These are very interesting for a whole variety of reasons that we don't have time to go into. But you should know that none have yet been approved, as far as I know, for use in humans, although they have been used in animals to date. And there's a lot of interest in this case, in the case of finding a vaccine for COVID-19, to try RNA and DNA viruses. Just before we go on, we have to think about why safety is more challenging when developing new vaccines compared to new drugs. When new drugs are going to be tested for bringing them to market, we're going to test them eventually in people who have the disease in question. So they're already suffering symptoms and some consequences of the disease. And then we have to consider the possible side effects of the drugs that might be used to treat the disease and determine whether or not the risk-benefit ratio is acceptable. In the case of a vaccine, to vaccinate a whole population, in this case, the population of the world, the vast majority of people who would get this vaccine will be healthy. And so what we would be doing is vaccinating people who by and large are healthy with a vaccine that may cause side effects. And even if the side effects are very rare, when you vaccinate millions or even billions of people, even a rare but serious side effect can affect a fairly large number of people. And so this is one of the reasons why we have to be particularly concerned when bringing new vaccines to the market. So where are we now? Well, after the early phases begin the first in human phases, the phase 1 and phase 2 trials. And you should know that there are already some vaccines for the COVID-19 disease in phase 1 and now in phase 2 trials. These are small numbers of people looking at the dose required, looking at the potential toxicities, and in the case of phase 2 studies, perhaps upwards of 100 or 200 people to see if the vaccine itself produces a robust enough immune response and if there may be evidence that it could work to prevent infections, although that's not the main purpose of phase 2 studies. If those are positive, we move to phase 3 clinical trials, and these are the pivotal studies aimed to answer the question, does this vaccine actually work? Does it prevent the disease, and to what extent? And is it safe? That is, are the side effects tolerable, and are there any serious side effects that we have to be concerned about? And with that information, the company or the organization that has developed the vaccine can go to the regulatory authorities and request approval to bring it to the market. And once it comes to the market, we then have to observe when giving a vaccine, again, to tens of millions of people or hundreds of millions, we have to observe after it comes to market what the potential risks might be, and is it as effective as we thought it would be based upon the clinical trials. So it's a lengthy process, but we've shortened it considerably to date, but the phase 1, 2, and 3 trials must be done. So where are we now? Much of the preclinical work, as I said, has been done, partly due to the fact that a lot of researchers were working on possible vaccines for the earlier SARS and MERS outbreaks, and partly due to the very new technologies that we have that are really quite remarkable. There are various types of vaccines that are now being studied as possible candidates, and a nice review article came out just a month ago in the journal Nature Reviews for Drug Discovery, and here's a slide that just shows what is in that paper. On the upper right part of this slide, you'll see 2 arrows pointing to some of the newer types of vaccines that I already spoke about that are being tried. These different colors just reflect the type of vaccine that's being used or being tested. And these are the ones that a month ago were in exploratory research stages, and these were already confirmed to be underway. And that was over 50 vaccines a month ago. It's even more now. And a month ago, there were about 35 or 36 potential vaccines that were exploratory but not yet confirmed. Some of those now have been confirmed. There were preclinical studies already being done in animals in a variety of these, probably 15 or 16 of these a month ago. These have moved forward. And even a month ago, there were 4 candidate vaccines in phase 1 clinical trials. There are more of those now. And some of these have already are exiting phase 1 trials and thinking about entering into phase 2 clinical trials. So things are actually moving along quite quickly. Phase 1 trials, as I said, have already started in some cases. The phase 2 trials depend on the results of the phase 1 trial to know what dose to give, perhaps how often to give it, although that is sometimes tested in a phase 2 trial as well. And that will lead to the planning and design of phase 3 trials. And these are going to be challenging. With any luck, we'll be ready within months to think about phase 3 trials, but these are going to be difficult for several reasons. Some of them are these. The infection rate of the population is actually diminishing, and that's a good thing. In BC particularly, as well as elsewhere in North America and Europe, the rate of new infections is declining. And if it is declined substantially by the time we have a new vaccine, it is going to be difficult then to get enough people in the study without going to many different centers in order to see enough infections in the control group to see whether or not the vaccine has actually reduced the rate of infection or the number of infections. So paradoxically, the better we do at controlling the infection rate, the harder it's going to be to do phase 3 clinical trials. There's going to be the question of what are the characteristics of the volunteers? If we test the vaccine all in younger people who are healthy and maybe at some risk for infection, we won't know if the vaccine works in older people or people who are sicker and have a variety of accompanying illnesses that put them at higher risk. So that will be a problem. And we may need international testing because we need a vaccine that can be tested in enough people to see if it works, and also across international boundaries to see whether or not it could be useful and could be marketed and distributed to various countries in the world. So we need cooperation to do this. So there will be significant challenges. Then we're going to need regulatory approval nationally and internationally. And then we're going to have to manufacture and distribute not thousands of doses, but billions of doses of this vaccine and make it available to everyone. That is going to be a huge challenge. So the new vaccine production and distribution process is a complicated one. We don't have to go into this now, but normally, as you can see down here, it takes almost 9 months to 2 years. So we have to try to be faster, and that's why people, including the Gates Foundation and others, are already putting money into developing factories, some of which that may never actually be activated if the vaccine candidates do not work. But people want to be ready to go if we do have viable vaccine candidates. So this process has actually already begun. The distribution is going to be the challenge, as I said earlier. So when will a safe and effective vaccination be ready for the general population? Well, the optimist would say, and this is just today, this came out early this morning, and this is Dr. Francis Collins, who's director of the National Institutes of Health in the United States, who was a leader on the Human Genome Project in the 1990s, and he is hopeful, he's the optimist, and he says we could have a shot by fall, but mass producing it will be a heck of a stretch by the end of the year. And lower down it says, if we get a vaccine, possibly by the end of this year, achieving a goal of 300 million doses, that would just be for the United States by January, will be a stretch. So he's the optimist. What about a realist? So this is Dr. Dale Fisher from the World Health Organization. He was interviewed and a story written about him over the weekend. And he's a professor for the World Health Organization. And as you can see here, the caption to the photo that was put online was that he rules out the possibility of making a COVID-19 vaccine before 2021. So I actually listened to that interview, and that's not what he said. What he said was this. He said at the end of 2021 is a very reasonable expectation because of the necessary phase 1, 2, and 3 trials of any vaccine to assess both safety and efficacy. So he's being realistic. He's not saying it's impossible. He's not saying he rules out the possibility of a vaccine before 2021, but he's saying it's not likely, and here's why. So when will we get such a vaccine? Well, this story came out early this morning on Reuters. Moderna, that is working with the NIH, has just announced that it has a potential vaccine candidate, which has just gained fast-track status by the FDA in the United States. And that means the FDA is prepared to give the reviews when phase 3 trials are done, a very fast track, hoping to get it to market if this virus vaccine turns out to be effective. And in that story, Moderna says that it expects to start a late stage study of the vaccine early this summer. And it says there is potential for a marketing application approval in 2021, an application approval. So they think they can bring a vaccine to approval by early 2021. Bringing it to market is another challenge. And I should point out that their vaccine looks very interesting, but it's an RNA vaccine, and to date, no RNA vaccine has ever been approved for use in humans. So we need to bear that in mind in terms of our optimism about this particular candidate. So once we get to the point of having possible vaccine candidates that are ready to come to market, there are questions we're going to need to answer even when the vaccine becomes available. Can a vaccine actually provide immunity? There's encouraging data from animal studies that it can. Studies in monkeys show that they can be made immune in the short term to this virus. How long will that immunity last? We have no idea, and we won't know until a vaccine is actually used and monitored in public over time. Which vaccine candidate would be the best? Well, we don't know. We won't know that until various vaccines have been tried in phase 3 trials, and we can use some of the data from the phase 3 trials to see which of those might be better than others. And therefore, we may be facing the reality of having several vaccines that come to the market and making decisions about which one we ought to receive. Will the vaccines protect the vulnerable in the population, those at highest risk? And we won't know unless the phase 3 clinical trials include robust components from that population, and I'm not sure that they're going to, but that remains to be seen. Will we be able to vaccinate everyone? That's the distribution question, and that's still an open question. It depends on the nature of the vaccine that turns out to be effective and how difficult it is to manufacture and distribute and to maintain in a viable state. So We have some questions that I have in my own mind about opening up the economy before a vaccine is ready, but I'm not sure we have time now. It might come up during the question period, so I'm not going to go over these now. I'm just going to thank you for listening and encourage you to ask any questions you might have. And I want to thank Aideen and Dan for their work in putting this together. They've done an amazing job. Thank you very much. Aideen?
Yes, thank you so much, Mark. And so I'm delighted to say that the questions are coming in. I'm going to switch over to the Slido platform right now. But I'd like to invite some of our viewers who may have arrived later to how to participate. The platform is in a tab in your browser. Simply type slido.com, SLIDO.com. And our event code today, again, is EXL512. Just take a moment for it to load. Okay, so Mark, can you see our question platform?
Yes, I can.
Okay, so I'm going to read out the questions to you. I'm going to highlight the questions, and I can see that there's a lot of questions coming in. So I'm going to invite our participants to if they see a question that they particularly want to respond to, just like it, and so then it gets pushed up to the top where I'll ask for our, it's set to a popular questions. So Mark, the first question is, do you think it's wise that things are going to start to reopen soon when we're likely not going to get a vaccine for at least a year?
So thanks for that question. It's a really good one. I was probably going to cover that in the last slide that I decided to pass over because I wanted you to have an opportunity to get to your questions. So that's a very difficult question. If we don't begin to open up the economy, it's going to take a long time. The soonest we're going to have a vaccine available to our population is sometime next year, I think, and it could be late next year. So that in itself is a problem. The question is, how long can we sustain closure of our economy? On the other hand, are we doing it in the best way possible? And there have been a few publications and a few discussions online that I think point in the right direction. But what it would take to open up things in the safest possible way is to have very large scale testing, both antibody testing to determine who has been exposed to the virus before we send them back to work, and an active and ongoing virus testing to see who is currently infected, and to determine in different workplaces when to isolate these people. And finally, we would also need contact tracing at a very high level to identify people in various work environments that have become infected, either asymptomatic but are showing positive virus tests, or are symptomatic and are showing positive virus tests. These people would have to be isolated once again, and their contacts would have to be traced in order to get them to lock down for a couple of weeks, just in case they have been exposed and have been infected. That's the best way to do this and do it safely and well. It looks like that's not going to be done here in British Columbia or in the rest of Canada, certainly not in the United States, and to some degree in Europe because they have greater experience, and to some degree in Asia. So do I think it's wise? I think it would be wise if we were doing it in the best possible way. Is it wise to do it in the less than the best possible way? It carries more risk, and we can talk about that risk if people want. But it can be done, but it ought to be done at the very least. If we're not going to do the robust testing and contact tracing that it takes, at the very least, we should have stringent efforts to protect the most vulnerable, because most of the deaths due to this virus occur in older people, as you all know, older people particularly who have a variety of coexisting conditions, and also in young people, most of whom, those who are seriously ill, are young people who also have coexisting conditions that put them at higher risk. We know who these people are, and if we can protect them to a great extent, then we can allow others to go back to work, knowing that they have a higher chance of being infected than they do now, and in fact, that's going to happen. The virus is going to continue to circulate, people will get infected, but if we do this right, we can minimize the number of serious infections and hopefully minimize the number of deaths in the absence of the robust testing that it would take to absolutely minimize those. So that's the best I can do on that question. Aideen?
Thank you. Would the vaccine provide protection while traveling to another country where there may actually be another strain?
That's a very good question too. So first, I think as I tried to say earlier, we don't yet know that the vaccine will protect us. That's going to come out of phase 3 clinical trials. The next thing is we don't know how long the vaccine will protect us if it turns out to have short-term efficacy. So that will take some time to know. And if it turns out that the virus is protective and it can protect us at least 6 months or a year, that's very good information. Then your question is, it comes to the fore, and that is, will it protect us against different strains of the virus that might have erupted or come to light in other countries? And that's ongoing research. We know that there are a number of different strains already of this virus. We don't know whether the vaccines we have will be effective against each of those strains. It depends on what part of the virus the vaccines target and whether different strains have mutated in a way that causes them to become less susceptible to those vaccines. That, I think we can be comfortable, will be monitored over time and in real time, because we have the technical capability of doing that. So we will get a tentative answer to that question based upon the viruses that are circulating in each country, but we won't know, of course, until people with a vaccine from Canada make the trip to Europe and see what happens.
So the trials will keep continuing, Mark, do you
what's called post-marketing research is going to be essential, as I tried to point out earlier. We need to answer these questions by studying the virus and by studying its movement through the population and its degree of protection and the potential side effects over time after the vaccine is given to the population. And that includes monitoring the virus for mutations and monitoring those mutations to see whether or not they would change the susceptibility of the virus to the vaccine.
Right. Mm-hmm. Next question, can COVID just disappear and not need the vaccine at all, and people develop immunity on their own? And the second part of that question, can COVID patient caregivers expect to have immunity?
Okay, so can it disappear on its own without us having the need to vaccinate at all? And we have absolutely no idea, but there's no evidence to date that it's going to disappear. We know that the earlier coronaviruses that were quite serious, the SARS and MERS virus, did disappear. They had very different characteristics, infectious characteristics, than this virus does. This virus is kind of in the sweet spot and seems to have the right characteristics to both maintain itself and yet not kill a lot of people, so that it can circulate in the population for a very long time. How long it can circulate before disappearing depends on whether or not, through herd immunity, either by vaccination or by infection, when most of the population does become immune, how long does that immunity last? If the immunity lasts for a prolonged period of time, then the virus could well dissipate and disappear from our surroundings, but that's as much as I can say on it. We won't know until enough time goes by for us to tell.
Thank you.
I think that answers that question.
Yeah, for sure. If and when COVID vaccine gets ready, should we expect that to be similar to the seasonal flu changing each year, or would that be similar to seasonal flu to implement?
So good question. Seasonal flu is interesting. Seasonal influenza is a virus that has never gone away. It's always with us. It infects us every year, and it mutates and changes every year. And one of the reasons it changes is because of the peculiarities of influenza vaccine. It moves back and forth quite readily between humans and animals. And sometimes genetic changes occur during that movement between animals and humans that can change the virus quite markedly. And so we have a worldwide surveillance program every year that monitors outbreaks around the world to identify which of the influenza viruses that is circulating is the one most likely to cause epidemics or even pandemics in the next year. And then vaccine manufacturers are alerted, and they use the same type of vaccine, but simply just altering the antigenic properties and the strain that they use to develop those vaccines. And that's why we're able to do that every year. We need to do it every year, in part because of these mutations and in part because of the duration of the immune response to the vaccine. With regard to the vaccines being developed to treat or to prevent COVID-2, we really don't know. If a vaccine turns out to be effective, we will have to monitor people over the first year and more to see how long that immunity lasts, and monitor the population for for local outbreaks of the virus to find out whether people who had been vaccinated are now being infected. That would give us an indication that we need to do something on a more regular basis, perhaps on an annual basis. But we won't know that until we get there.
Okay, thank you. Do you think people will be reluctant to get the vaccine right away since it's so new?
Well, I'm hoping not. I think that it will depend somewhat on the number of people in the phase 3 clinical trials that use vaccine to the market, and the data being released and reported from that trial. Typically, 3,000 to 5,000 people or more will be treated in a phase 3 clinical trial. In this case, we may need even more because of the declining infection rates. So if 5,000 or more people are treated with this vaccine, and of course, whenever these clinical trials are done, it's essential that very careful monitoring for adverse effects of the treatment be recorded and documented. And so we will have fairly good information over a period of months in a group as large as 3,000 to 5,000 people. Are there any serious side effects, and what are they? What are the more common side effects, and how bad are they? And we'll be able to explain to the population, this is what you can expect. These are the people who were vaccinated. This is what happened to them. And this is what we have or have not seen in the way of serious side effects. And then the rest will be up to each of us to decide if we want to take that vaccine until more information is known. We know that there are people in the population who are against the idea of vaccination itself. And so those people are very unlikely to take the vaccine regardless of the data we get from phase 3 clinical trials. And that's unfortunate, but that's just the way it is.
Okay, thank you. A question about some trials at UBC. Have you heard of the new drug trial led by UBC's Dr. Josef Penninger? The drug is supposed to block the ACE-2 receptor from binding to human cells.
Yeah, thanks for that question. So Dr. Penninger's study would have come to the UBC Clinical Research Ethics Board, which is one of the 6 UBC-based research ethics board. Our ethics board at Children's and Women's have not seen that trial, so I can't speak to it. I have access to it, but I have not seen it, nor do I know whether or not the details of that trial can be discussed in public. So I can't say much more about that trial right now. If you have a specific question about a specific drug, I can say, by the way, that the idea of blocking the receptor is an interesting one. And I mentioned earlier during the talk that one of the ways to try to block infection is to block the receptor that the virus uses to gain entry into the cell. The ACE-2 receptor is the receptor that allows the virus to gain entry. It's kind of like putting your key in the lock of your front door, turning that key to gain access to your house. If you don't have a key that fits, you can't get in. And the virus needs to have an ACE-2 receptor to unlock the way into a cell. And so what Dr. Penninger's study is trying to do is to block that receptor so that the virus, when it gets to the cell, cannot unlock that lock.
Thank you for that simplified explanation. I appreciate that. Do we have good methods to compare multiple vaccines in the global trials? And then a second part, would there be multiple vaccines in the market, or one of the best globally?
Well, that's a very good question and not an easy one to answer. Do we have good methods to compare multiple vaccines in the global trials? Right now, no. But we do have good methods. When studies have been done and done well, when the data are published, we have good methods for comparing results of various different trials when they're designed in a similar fashion, looking at a similar result. So it's interesting that you raised the question, because once again, just in this morning's news, I discovered that there is international work being done to bring people together who are working on different types of vaccines, to encourage all of these groups to design their phase 3 clinical trials in very similar fashions, to use similar measures of outcome and similar timescales and similar populations to be studied, so that when we get results of more than one vaccine, we can look at those and draw some comparative inferences from those initial studies. Beyond that, it takes many more studies and statistical techniques to do real statistical comparisons of the benefits of different vaccines. So that would take a long time, but we can do fairly well if the international consortium, this unofficial consortium, agrees to design their studies in a very similar fashion. And so I'm hopeful that that's actually going to happen. Would there be multiple vaccines in the market, or one of the best globally? So we're in a race. Everyone's in a race to get a vaccine that works, and we don't know which one or which ones are going to work. A number of these are going to come to clinical trials over the next 6 to 12 months. And I'm hopeful that more than one vaccine is going to work. And probably if one is found to work, the question is, well, how well does it work? Does it reduce infection rates by 20%, by 50%, by 90%? We won't know until we see the data. But if it's got an effect of perhaps as good as 50%, well, that's better than no vaccine at all. And that vaccine is going to be approved if it's safe enough. Other vaccines may come along that appear to be more effective and therefore could actually prevent a higher proportion of infections. If safe enough, those vaccines will also come to market. And then we're going to all need to make decisions about which vaccine or which vaccines to take, or should we take more than one vaccine? This story is going to evolve over the next several years, I think.
Mm-hmm. Thank you. What are your concerns about any side effects related to a fast-track vaccine, Mark?
So again, these are great questions, by the way. Thank you all very much. So that question actually was discussed in part of the article that brought to attention the fact that the FDA is now going to fast track this first vaccine by the Moderna NIH consortium. And the goal is, of course, to have as rigorous a phase 3 clinical trial as we would always have. So we ought to have, from the phase 3 trial, we ought to have the best data that we can possibly get from one study of the side effects of the vaccine. I think the data will be robust with respect to side effects, but what we won't know is, are there more rare and more serious side effects? If a side effect occurs in 1 in 5,000 cases and only 3,000 people have been vaccinated in the phase 3 trial, we may not have seen a case. This happened once in 1976 when the swine flu was a great fear. We thought there'd be a great pandemic of swine flu, and a vaccine was rushed to market, and it did not have adequate testing. And it turned out that swine flu didn't turn out to be a serious pandemic in that year, but millions of people did get the vaccine, and several hundred people did develop a fairly severe neurological side effect, which couldn't have been known from the rapid studies that were done. So again, bottom line is, I think the studies will be done as carefully as they always would be. But how many people will have been studied, and how comfortable or confident can we be that serious side effects are unlikely? That depends on the numbers, and we'll have to see that once the data are published.
Mm-hmm. And were those disorders permanent after within the swine flu?
Guillain-Barré is often temporary. Some people can recover but be left with some neurological deficits, but sometimes not.
Right. Next question. Are there estimates of how much rushing out a vaccine will exacerbate vaccine hesitancy?
That's a very difficult thing. I haven't seen any estimates of what the rush to get through the phases 1 to 3 will do. I think we'll only know that when a vaccine comes to market. I'm guessing that there will be a segment of the population that will not want to be vaccinated regardless, because they're against vaccinations. There'll be another segment of the population that's going to be very concerned about taking the vaccine if they don't feel that enough is known about potentially serious side effects. And they may forego initial vaccination, or maybe the vaccine is not as effective as they would have hoped, and they're unprepared to take the risk that might be involved with unknown side effects for a vaccine that's less than 90% effective. And so there'll be a segment of the population who probably will not want to initially be vaccinated, and I have no way of knowing how large that segment will be. And there'll be a very large segment of the population who will want to take a vaccine that has been shown to be effective to some degree at reducing infections with a degree of common and not very serious side effects, and no strong evidence of serious side effects in a group of 3,000 or more people. So I think there's going to be a mix of those 3 large groups in the population.
Thank you. Are there people who have natural immunity, and how can we test for that?
Wow, that's a very good question. So I don't know the answer to that question. There is such a thing as innate immunity that we have as a consequence of being infected with other viruses or being treated with other vaccines, like the BCG vaccine for tuberculosis, that seems to trigger the innate immune system that might confer some degree of protection. There is one Nobel laureate chemist who actually believes that there may be some natural immunity in the population. And he argues that this is the case based upon his study of the virus outbreak in China, Korea, Taiwan, and then eventually in Europe. And he saw a degree of decline over a period of 4 to 6 weeks in the infection rates in countries that have varying degrees of lockdown. And he was asking himself, why would rates be declining even in countries that did not have much in the way of isolation or lockdown? And he thought that might be evidence that there is growing immunity in the population as well as natural immunity. So that latter part, the natural immunity, remains an unknown. The growing immunity part was based on the idea, which is still circulating, that if we can allow enough people to be infected, then we might have what's called natural herd immunity by infection. That means that if immunity to reinfection occurs when we get infected with this virus and get the disease, then if enough of us become infected, then the virus cannot move so quickly through the population. And it's been estimated that it would take between 60 and 80% of the population to be infected in order to confer herd immunity on the population. So it's very clear to me that a safer way to confer herd immunity would be vaccination, and not to allow 90% or 80% of the population to actually become infected by the virus.
Mm-hmm. So next question, Mark, why is there so much focus on drugs when they are promising herbal and supplemental treatments that have come out of China and Europe?
Okay, so this raises a number of important questions. So when asking the question about promising herbal and supplemental treatments, so the question is, what does promising mean? And what do they mean by herbal and supplemental treatments? So we have to differentiate when treating any disease, what we are treating. Some drugs and some non-drug remedies are meant to treat symptoms of a disease. If people have aches and pains and fevers and sweats and other discomforts, we often have drugs and other treatments that are meant to make people more comfortable, to treat the symptoms, but they do not eradicate or shorten the disease, nor do they prevent the serious consequences of the disease. I know of no promising herbal or supplemental treatments out of China or Europe or elsewhere that show any evidence of treating the disease itself. If there are those that may mitigate some of the symptoms, some of the discomfort that people have when they have the disease, and people want to try these, they're welcome to do so. But we also have no safety information. So it is quite possible that some supplements and some herbal treatments can actually adversely affect one's response to the virus. And without more information, we cannot possibly know. Some of the drugs that people wanted to try, some of the treatments people wanted to try because they thought they might be effective, turn out to actually, when used in people at high or low doses, make people more likely to get a more serious infection than others. So one has to be very careful. I would be very cautious about using unproven but so-called promising treatments that have not undergone rigorous testing, especially in the presence of a new virus and a new disease. I hope that answers the question.
Mm-hmm. And I'll just let this listener know that on May 19th, we do have a lecture which covers the topic of integrative health systems and what it's offering us during this COVID pandemic. So that might be of interest to people who are interested in that particular topic. Is there any active research, Mark, happening on natural treatment supplements for COVID-19? So I guess that's quite similar to what we just discussed, or?
I can't answer that question, but you've already seen how difficult it is to mount well enough designed and large enough studies to see if drugs will help to treat the disease. And again, the question is, what do we mean by treatment? Do we mean shorten the course of the disease, reduce the mortality of the disease, or to help treat the symptoms of disease? So that would need to be clarified in the question. And if we could interact with one another, that's what I would ask you. But I know of no current research. And I guess given the urgent nature of the condition, I think all the attention is going to be paid on treatments and in particular on vaccinations that have the hope of getting us out of this pandemic situation. And probably it will come later that we have people who will be willing to spend the money, the time, and the effort to see whether or not so-called natural treatments can mitigate the symptoms of disease or possibly affect the course of the illness.
Okay, thank you. I've heard COVID affects men and women differently. Does this affect how the vaccine is developed, and are clinical trials separated by gender?
So, yeah, boy, excellent question. So it does look like men and women are affected differently. I'm not sure of the reason why. I know there's some research being done now to try to understand why. Does it affect how the vaccine is developed? Probably not. But will clinical trials be separated by gender? I'm almost certainly not. There should be clinical trials that will include both all genders and also include people with other or comorbidities, other existing illnesses. And this information, when people are enrolled in a clinical trial, needs to be very carefully documented. And at the end of the trial, when the data are analyzed, it is possible to determine whether gender made a difference, whether pre-existing condition like coronary disease or heart disease made a difference, or asthma made a difference. So it depends on the population that's initially going to be studied in the phase 3 trial. But I'd be very surprised if both men and women were not included in those phase 3 clinical trials. In fact, it would be shocking to me if they weren't. So I think they will not be separated by gender.
Mm-hmm. And actually, again, another lecture coming up on our last lecture on May 26th is what can gender data teach us about COVID-19. So again, that may be of interest to yeah. Which country do you think is most likely to develop an effective vaccine?
So that's a best guess question. Best I can say is that the British are very close. They're in the phase 1 stage of an interesting trial vaccine. There are 2 or 3 candidates in the United States. They're also looking very close, and there's some in Europe that look close as well. Which of these is going to be first? That's actually a biological question. They're all getting very close to actually getting into or are already in clinical trials. They will get to phase 3 clinical trials, but we have no way of knowing now which of these vaccines is going to work and be safe enough to administer. And this is a biological question. It's an empirical question. We won't know until it's tested in people and in enough people. So I can't say which country is going to be first out of the blocks. Sorry.
Okay. Mark, do you think there'll be a vaccine that will be made mandatory for kids to be able to attend public school?
Mandatory for this virus? I don't, do I think that, so first of all, the vaccine is not going to be tested in children initially, because you can't really test a vaccine in children in the first instance unless it's intended for children and unless it meets certain criteria. So children are not likely to be in the first order phase 3 clinical trials. I'm not certain of that, but I don't think so. But once the vaccine comes to market and it's shown to be effective, and if and when it's tested in children, which probably it will be, and if it turns out to be effective in children, then your question is, do we think that it will have to be mandatory for kids to be vaccinated in order to attend public school? There are just a lot of questions we need answers to before we can get there, right? You can make the case, there are reasons why, not so much for the children and infecting other children, because although some children can become seriously ill with this virus, it's a very small percentage of the children, very small percentage. And then the question is, well, can kids pass the virus on if they do become infected to their parents or their grandparents or their neighbors and friends? And that is, are they basically low symptom or asymptomatic carriers of the virus who can transmit it to others? And we don't have enough information yet to know the extent to which this happens. When we have more robust information, we will know how important it is going to be to have children vaccinated. And we're just not there yet, I think.
Okay. And so, Mark, I'm conscious of time. It is gone 2 o'clock. Are you okay to do a few more questions?
I'm fine. If you are and others are, yeah. Yeah.
Okay, thank you. Aside from the possible dangers of a fast-tracked new vaccination, if one did turn out to have dangers, that would lend credibility to the anti-vax people. Comments?
If everybody in the population were focused on the idea of making decisions based upon the best possible evidence that we have, it ought not to make a difference. We have many drugs that come to market that have very serious side effects. And we need to understand those side effects and decide who is going to be a candidate to use these drugs. Now, some of these drugs that have very serious side effects are used to treat cancers. Some of these cancers are life-threatening. some of the drugs are quite harmful, but if it can cure a patient of a cancer that is very likely to take his or her life, and the side effects, severe though they are, can be mitigated and managed in some way, then each individual has to make a decision. Do I want to enter into a therapeutic trial with my doctor to take this drug that has some serious side effects, knowing that I have a disease that is also very serious and could kill me? On the other hand, we're talking about a vaccine in this case that is going to be used for healthy people that might have serious side effects. So each of us has to look at the evidence, not at what some people think, what is promising, what is of concern, or what they believe about vaccines in general as being a bad thing to do. But what about this vaccine and this disease? How likely is it that I'm going to become infected? And if I become infected, how likely is it that I will be seriously ill or I will make others seriously ill? Once I have an estimate from the data, from the evidence about those probabilities, then I can say, okay, if I do get myself vaccinated, what is the probability that I suffer side effects? Well, maybe 10% chance of headaches or swelling or soreness or even a fever that will go away within 48 to 72 hours. Well, that's not so bad. What if I have a 1 chance in 100 of having a severe neurological problem? Well, that's a concern. I'd have to think carefully. What if I had a 1 in 1,000 chance of having a severe side effect? That's less of a concern. What if it was a 1 in 10,000 chance that I'd have a serious side effect? That is a less serious concern. The fact is, we all face risks in our lives. Every time we go out for a drive, every time we get on an airplane, every time we get on a boat, every time we cross the street, we're taking some risk. We don't normally think about it that way, but in fact, that's what we're doing. And when we vaccinate ourselves, we're taking some risk, and we have to understand the best evidence for the degree of risk and the seriousness of the risk, and balance that against the possible benefits of doing what it is we want to do. And so without looking at the evidence, without listening to reasoned and knowledgeable people who explain clearly what we know, what we don't know, and what we believe to be true with the limited evidence we have, in order to help each of us make our own decisions. That's what we need. For people who rule out making evidence-based decisions for their own care and that of their loved ones, there's not much more I can say.
Mm-hmm. Okay, so there's a lot of questions still coming in. So I'm going to start moving through them and look for questions that are a little bit different. Okay. So once we have a vaccine, Mark, do you think things like social distancing and wearing masks will still be implemented?
So that's a good question. If we have a vaccine that has been well trialed and is brought to market with robust data from a phase 3 clinical trial that includes important segments of the population, and if the data suggests that this vaccine is capable of reducing infection rates in the order of 70% or more, and among those who do get infected, if the evidence is that the severity of the infection appears to be less as well, I think that there will be a much poorer case to be made for social distancing and isolation if enough of the population choose to become vaccinated. And that means 60 to 70% or more of the population. If they choose to, and if we have the availability of the vaccine to vaccinate that number of people, then I don't think we will need to undergo isolation techniques at all, except for those at highest risk who perhaps cannot be vaccinated for some reason, or who don't respond to the vaccine for some reason.
Okay. Any advice or any knowledge about people of particular races that are affected by COVID? This person has heard of the African descent.
Yes, I've read some of this information myself. I've read no scholarly articles on this question, but the best that I've read, it would make sense. If these are African Americans, people living in the United States of African descent, if they are from the as many African Americans do, such as type 2 diabetes, high blood pressure, and obesity and other diseases or illnesses or characteristics that put them at higher risk. So these are people who, by definition, not because they're African Americans, but because of their social circumstances and their health that is related to their social circumstances, this is what is putting them at greater risk. Okay. How can we ensure that the vaccine will be effective across races? That gets back to the issue I raised several times, I think, already. The phase 3 clinical trials that we will use to make decisions about bringing a vaccine to market will be the answer. It is what segments of the population are going to be included in those phase 3 clinical trials. For example, if those were run for some reason in a way that excluded African Americans or African Europeans or African Canadians, then we would not know. We would not have an answer. And therefore, we'd have to say to those people in the population, we don't know what this vaccine will do for you or do to you. Okay.
Do you think vaccines could possibly be made mandatory for international travel?
That's another very good question. I think the answer to that one, this is just my personal sense. I've not thought about this yet, but I think the answer is yes. I think that if we have a vaccine that is demonstrably effective, such that it reduces the carrier state, that people who might be asymptomatic but carrying the virus, and it induces active infections by the virus, and that's been well documented and the evidence is strong, then I can readily imagine that North American countries, European countries, and Asian countries will say, you will not be allowed into this country unless you show evidence of vaccination. It's already the case that there are some countries you cannot travel to unless you show evidence of vaccination against yellow fever, for example, and perhaps other illnesses. So yes, I think this could well happen as long as as long as the SARS-CoV-2 virus is with us. Yes, I think we will probably need to show evidence of vaccination in order to be able to travel internationally. That's my guess.
Okay, 10 minutes past 2, and I know we're coming to the end, so I'll do 2 more questions. Okay. Affordability. Any sense about the affordability of a vaccine, Mark?
I think that's a countrywide question. It depends on whether the vaccine is going to be paid for by the healthcare system in whatever jurisdiction people live or not. As to the affordability, that's a tougher question. We do now have growing agreement amongst the consortium of people working internationally, testing a variety of types of vaccines. I'm hoping that there will be international cooperation on the marketing and distribution of vaccines to keep costs down from that perspective. And then the question is the risk reward for the sponsors of the development of the vaccines. How much will they want to charge for their vaccines? How much will governments be willing to pay? And that's going to depend on a jurisdictional basis. I know there's a huge debate just started up recently, and I've seen it online just this morning, in the Senate of the United States. Should the government be paying to vaccinate people once a vaccine comes to market? My guess is that they won't. In Canada, my hope is that they will.
Okay, thank you, Mark. And I'm so sorry, I'm going to skip this question. I'm going to jump to this one because it's a little bit different. Okay. Oh, would you believe it? I think I answered it instead of highlighting it. It was regarding the US. Oh, sugar. I can't quite remember. It was something to do with, it was around the US hoarding. If the US was to hoard the vaccine.
Okay, so you notice in this story that I saw online and I put in the slide, Dr. Collins was saying he's very hopeful that we will have proof of principle for a vaccine and maybe even a completed phase 3 trial by the end of this year, and that we could perhaps get filing for approval in the United States early January. But he thought it would be a real stretch to get 300 million doses available in a reasonable period of time. That tells me that he's thinking about the United States, right? So each country is now thinking about which vaccine is going to be effective. And someone asked a question about which country is most likely to produce it. That's very interesting in this context, because whichever country produces it is going to want to vaccinate its own people first. And if it's a vaccine that can be produced in the thousands or millions of doses, it's not going to be circulated around the world. It's going to be circulated within the country of origin. So we won't know until we know which vaccine or vaccines are successful, and how easy and inexpensive they are to manufacture, and how widely distributed they might be able to be made.
Mm-hmm. And that was, the affordability. And is it likely that international pharmaceutical companies would come together if there was one vaccine that was proving very successful?
In terms of manufacturing?
In terms of manufacturing.
Yeah, I mean, I didn't know much, but I saw in this story online that there's a willingness to think about doing this. Okay. And I have read several times, haven't seen Bill Gates speak about this, but the Gates Foundation is putting a substantial amount of money into developing manufacturing capabilities for vaccines that would require a variety of different techniques, a variety of different platforms. And knowing that only one or two of these might be successful, they could be sinking millions of dollars into ones that will not work. But they're willing to do that in order to be ready when the vaccine does come to market. So that's hopeful.
Right. Okay. So my apologies to that one question, and also to all the other people with their super questions that we didn't get around to today. And so now I just move on to the thank you. Thank you, Mark, so much for that fabulous presentation. I certainly feel like I've learned a lot today through your lecture, but also through the great questions that came in.
Thank you, Adine. I really appreciate it. And certainly all the participants, those questions were fantastic. And I hope I helped somewhat to give you some information that you didn't have when you first joined in. So best wishes to everyone.
Thank you. So thank you to our listeners for making us part of their Tuesday. Just to remind you that the recording will be available on our YouTube website very, very soon. And if you need to contact us, our contact details are info.exl@ubc.ca or phone 604-822-144. Goodbye, everybody, and hope to see you soon, and stay safe.
Thank you very much, Adine. Thank you,
Take
Dan.
care. Bye-bye.